So you might know that vertebrate development has mainly been studied in these four models: mouse, chicken, frog, and zebrafish. To study development in these animals, CRISPR has been used as a powerful tool to edit DNA and modify gene function. CRISPR has also been adopted in many other different vertebrates. But did you know that there isn't a great model organism to study gene function in reptiles? There are over 10,000 different species of reptiles, but none have been well established as models to study development and genetics. Our lab set out to find a new model to explore reptile biology. We looked into a group of lizards called Anoles. These lizards have been well studied to understand reptile evolution and ecology, and they're often compared to Darwin's finches. Anole species show incredible variation in their color patterns, body structures, sizes, and behaviors. One of the biggest differences you'll see between anole species is limb length. Lizard limbs are adapted for the specific microhabitat that the animal lives in. For example, the Cuban twig anole has tiny little limbs that allow it to better cling onto thin twigs and trees. Another lizard, the stripefoot anole, is a type of trunk ground anole. It uses its long limbs to run and jump between different surfaces at the bottom of the forest. Interestingly, differences in limb lengths between species are observed very early in development, well before lizards hatch out from the egg. To understand how limbs have evolved in different species, scientists have compared genomes of long-limbed versus short-limbed anoles, and they found regions that may be controlling limb length. But there hasn't been a good way to test the role these DNA sequences play in limb evolution. To study reptile genetics, we developed a method of gene editing in the brown anole, Anoles segri. These lizards are invasive in the US and are commonly found throughout much of the southeast. To study these lizards, our lab travels to Florida several times a year to catch them. Yes, you can use your hands or a net to grab them, but another great way is to use a tiny little lasso. Wrangling lizards is a lot of fun, but there are other reasons why they make a great developmental and genetic model. First, the brown anole is small, and not surprising given that they are an invasive species. They're easy to raise and they thrive in the lab. Lizards are fertile most months of the year, and one female can lay 1 to 2 eggs a week. Eggs take only about a month to incubate before hatching. And the many embryonic stages of the brown anole have been well described by different researchers. So how do we perform gene editing in the brown anole? It's not so straightforward because reptiles have a unique reproductive biology. Lizards undergo internal fertilization, and the time that this occurs cannot be predicted. To overcome these challenges, we use a new method to surgically access the oocytes, or the unfertilized eggs, so that we can inject them with CRISPR gene editing components. Next, we seal up the lizards and allow them to recover. We wait for the oocytes to become fertilized by sperm and eventually develop and be laid as eggs. Using CRISPR, we decide to create a mutation in a gene called tyrosinase. This gene is necessary to form pigment, and mutations lead to albinism in other animals. Interestingly, humans with albinism often have vision problems, and this is due to underdevelopment of a part in the retina we call the fovea. This structure is important for our clarity of vision. Until now, foveal development has not been very well studied because none of the major models used today have a fovea. But the fovea is present in anoles. After injecting dozens of oocytes, our team was able to make gene-edited albino lizards. To our knowledge, this was the world's first genetically altered reptile. Because lizards have a fovea, we now have a model to study eye development and better understand how visual defects occur in humans. Furthermore, we believe the gene editing strategy we have used in anoles can also be applied to other reptiles. There are still many unanswered questions about reptile biology and evolution. Using CRISPR can pave the way for researchers to study the genetic, cellular, and molecular basis of development in reptiles. As Dr. Sanchez Alvarado highlights in his inspiring TED Talk, we can solve the oldest and most challenging problems in biology by studying new research organisms. I'm Aaron Alcala, a PhD candidate in genetics at the University of Georgia. If you have any questions about any of the topics covered today, please feel free to contact me. Thanks for watching.